Welcome back to Unit 4, Lesson 2, Part 5. Now we're going to talk about a very specific example of microtubules called the centrioles. And the centrioles are these little perpendicular structures. So there's two, they always come in pairs. They're in a perpendicular format. And what we see here is an electron micrograph of, this is viewing one of them end on, this is viewing one of them sideways, because as I said, they're perpendicular. And in cells that are dividing, you will have two sets of centrioles, one on each end, that help pull the chromosomes apart. They have a very recognizable structure. Centriole structure is there are two pairs at time of cell division. Located at opposite poles of the cell. And they are made up of what we call 9 triplets of microtubules. So if you go back up here and look, and you have to look kind of carefully at this diagram. I'm going to erase this part. We'll highlight it again, but in more detail. If you look really carefully, you can see there's this pattern here where you have 1, 2, 3 microtubules. And then again you have 1, 2, 3 microtubules. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2. The third's hard to see there, but I guarantee you it's there. 1, 2, 3. 1, 2, 3. 1, 2, 3. And if you count, that's 1, 2, 3, 4, 5, 6, 7, 8, 9 sets of triplets. So somewhere in here is a third. It's just kind of hard to see on that picture. Okay? And each part of the centriole is always made up of that pattern. It's very clear to see under the cell, or under the microscope. So, function-wise, they attach to spindles, which attach to chromosomes, to pull the pairs of chromosomes apart. During cell division. Make sure that one chromosome goes to one side and one to the other side, so that each new cell gets one copy of the chromosome. And if we look at cell, it's like a car factory. And centrioles are like winches So they'd be the actual winch, and the spindle fibers would be like a rope or a cable attached to whatever was being pulled apart. Now, all of these things, apart from the nucleus that we've been talking about, are actually suspended in the cytosol. And you're going to encounter the use of the word cytosol and cytoplasm, and sometimes it's used correctly, sometimes they're used interchangeably, but they really do mean different things. So the cytosol, and I haven't drawn it here, if I color this, I will make it a real big mess. So the cytosol is all of this area that everything's suspended in. And I am not going to color it because I know it's not going to turn out well using these pens. Okay, so they're not pointing to anything in particular. They're pointing to all that clear space. Okay, so cytosol is the gel-like material. Between the nucleus and plasma membrane. Organelles, proteins, nutrients, and waste are suspended. Say suspended in the case of organelles, or dissolved in the case of nutrients. It's where most metabolic reactions of the cell occur. Now the cytoplasm, as I said, many people will use them interchangeably, but if you were told as a scientist that someone would like you to prepare cytosol or cytoplasm, it'd be a very different procedure. Okay? Cytoplasm refers to everything between the nucleus and the plasma membrane. So the gel and all the organelles. So if I say that ribosomes are in the cytoplasm, I would be implying that they're also found inside the organelles, which isn't necessarily true. So there is really a difference to using the terms. We go to our analogy. The cell is like a car factory. Then the cytosol is like the air in the factory. It surrounds everything, supports everything that is occurring. And finally, we've worked our way from the inside of the eukaryotic cell to the outer edge and have reached the cell membrane, also known as the plasma membrane. It's important to typically use the term plasma membrane because cell membranes can be from the nuclear envelope to that forms the Golgi, the mitochondria, and they sort of vary throughout. We're going to look at specifically the plasma membrane. So, we can color it up here. plasma membrane, we're going to talk about this portion of the cell. As you can see, there's two layers to it, and this is a close-up diagram. And the fancy word for inside of the cell, blank cellular, is correct, intracellular side. So this would be cytosol beneath it versus the outside, extracellular. And the plasma membrane has a very complex and dynamic structure. We're going to abbreviate plasma membrane, PN. So it's complex, dynamic, but it has a consistent basic structure. So it's made up of a vast variety of proteins embedded within the phospholipid layer. And remember, it's a bilayer due to the polar nature of the phospholipid. Standard model of the plasma membrane is called the fluid mosaic model, which perfectly describes the nature of the plasma membrane. Fluid because it's dynamic, changing. It isn't solid. Things flow around in it. Seems kind of weird to us because on the macroscopic level, our cell membranes, which make up our skin, feel very solid to us. But on the microscopic level, they're actually fluid with things moving about. And mosaic is a fancy word meaning mixture because it's a mixture of all types of lipids and proteins. So it's a very good name for the model. We're going to look down here and take a closer look at some of the details. And draw it. First, we're going to draw our phospholipids. We're going to color code our phospholipids. The phospholipids have heads just like water. And then they have tails that fear water. They're non-polar. They don't like interacting with the polar bonds of water molecules. And remember the structure of your phospholipid. You've got your phosphate group, the head group, which is polar, attached to the glycerol backbone, and also attached to that glycerol backbone are two fatty acid chains that are nonpolar, and that's the red I've drawn. So, polar, phosphate, water-loving. Here, non-polar fatty acid pairs, which are what? Correct. Hydrophobic, meaning they fear water, and that's why they're hiding in the middle of the bilayer, away from the water. And just to remind you the details, all together, this is called a, and because it has a polar end and a non-polar end, it's said to be bipolar. Another word you might encounter next year, anti-acid, which also means bipolar. Okay? And this is considered the whole thing a lipid bilayer. So as I said, embedded in it and attached to it are proteins. We have proteins like this. You can have proteins. A little bit more of the bilayer. Some proteins. This. I'm going to call this A, B, B. So we can clarify what they are. So change the color to black. A is an integral membrane. Or intrinsic. So use both words. What that means is that spans the plasma membrane. So it goes right through the plasma membrane from the extracellular side through the lipid bilayer into the intracellular side. Integral plasma membrane proteins have to have a hydrophobic region in the middle just like similar to the phospholipid. B is what we call a peripheral. Think about what peripheral means. What's your peripheral vision? That's right, the ability to see to the sides. Well, B is called peripheral because it's on the inside or outside of the plasma membrane. They aren't right across. So it's a peripheral, or because it's on the edges, not the inside, extrinsic membrane protein. So it's somehow attached to that membrane. Sometimes they actually have little tails, fatty acid tails, that help embed them into here. And because B is on the outside, it is considered extracellular. In contrast to C, which again is also considered a peripheral or extrinsic membrane protein. These ones are intracellular. So what I've just described here is sort of three types of structural plasma membrane proteins. But there's actually 5 sort of different functional classes of plasma membrane proteins. 5 functional classes of plasma membrane. So we just represent, as many pictures do, the plasma membrane just as 2 lines. So this is the plasma membrane. We'll call this the extracellular side and the intracellular side. We have, as I said, 5 key functional classes. First one, I'm trying to give you the impression three-dimensionality. And it looks like a barrel. And that's not a very good job. Let's try that again. So the 5 functional classes of proteins, or membrane proteins, are as follows. What we're going to do is we're going to draw a representation of the plasma membrane. And like most people do, can't draw all the phospholipids in the tails. They just simply draw 2 lines representing the bilayer. Again, the extracellular side. Here, intracellular. Okay. First one typically looks almost like a barrel. almost center. And it allows things to pass through the plasma membrane as they travel through the barrel center. So things can travel through this and out the body, or in the other direction, depending on it. So this one's called a channel protein. The next one, a little bit more difficult to show. It does have a channel through it, but the channel isn't constantly open. So often it's represented to look like this. And then plus. And that's just showing it over time. As time progresses, it changes. This is called a carrier protein. I'm not sure if you caught that beep or beginning of that PA announcement, but that's typically why I don't film at school, because I don't want those getting picked up. Okay, the next one, they have very specific shapes. And their shape can change when something binds specifically to it, and they can signal to the inside of the cell. These are called receptor proteins. The next one I'm going to actually color green, though you can't expect on a test it to be coded green. But you guys knowing my obsession at this point, what do you think this protein is going to do? If you guess that it was some sort of enzyme, you have it correct. So this type of protein is an enzymatic protein. And the way you can tell it in this diagram is that there'll be a little picture indicating time, and in will come something change its form, because some sort of enzymatic reaction has to be happening. So it becomes something else. And finally, we're just about out of space, but we'll make it work. Ask this one off of it. glycolysis chain. Off of it is a carbohydrate chain. And these are called cell recognition. Wow, I really have to squeeze that in there. Hopefully you have a little more space on your page. So cell recognition proteins. And because they have a carbohydrate chain, we call them also glycosylated proteins. Not all glycosylated proteins are cell recognition proteins, but most cell recognition proteins are glycosylated. Let's talk about what these things do. The first is your channel protein. And it has a hollow channel through the middle. It's a tunnel. Like the tunnel that goes under the English channel. That's how I remember it. The channel. Tunnel that helps charge the molecules. Things that are cleaner or ions pass through a membrane. Because if you think about it, if it had a positive charge and the polar head's a negative charge, it would get stuck. It would stick right to that. And if it had a negative charge, and the phosphate polar head's negative, it would just bounce off and it won't be able to go through. So what we need is a nice little protected space through a protein channel that will allow these ions to flow straight through. The next one is called a carrier protein. And these specifically bind to molecules and transport them across the membrane. So, whereas the channel protein sort of like any ion that's small enough to pass through it, the carrier protein is very choosy about who it carries. So this is like a tunnel. Any vehicle can go through the tunnel. This is like a ferry where if you're too big, you're not allowed on the ferry. You have to be the right size. Very prominent example of this that we're going to look at when we get to the nervous system is sodium. Na stands for sodium. And potassium. K stands for potassium. Channels. Or I should say, not channels. Sorry. Pumps of nervous system. And the reason for this being this picture up here, what it's trying to show is that it will bind to something on the outside. It will cause the receptor to change its shape. And then it will essentially spit it back out on the inside. So that's what that picture is trying to show. Why there's two of them. It's the same one, just changing over time. The receptor protein acts like satellite dishes. Receiving signals transmitting it to the inside. So transmitting info into cells. And again, this is going to be relevant in the nervous system when we look at neurotransmitter receptors. Definitely spelling that wrong. Neurotransmitter, all one word, receptors. Enzymatic proteins, well, these are located all throughout your cell, but some are specifically associated with the plasma membrane, and they simply are biological catalysts that speed up specific reactions. Finally, our cell recognition proteins are unique to our cells. So my glycosylation is likely different from your glycosylation. And they're basically like when your mom, when you were a kid, put a name tag in your clothes. It's like a name tag on our cells saying you belong to this body. And anything that comes in without that name tag is determined to be foreign, and your immune system tries to attack it and shut it down. Name tag. IDs cell as cell. Wrong tag. Something comes in with the wrong tag, the system attacks. What happens in transplant rejections? What happens is the donor has different tags. The recipient and the recipient's body attacks it. One of the ways we try and prevent this is we actually suppress the immune system of the recipient if we can't get a perfect match. Okay, so if we look at the overall function of the plasma membrane, it separates inside a cell from external environment. It monitors and communicates extracellular conditions to inside a cell so the cell can respond. And it regulates what enters and exits the cell. Because not all things can enter and exit, it's termed, the plasma membrane that is termed semi or selectively permeable. Next day we'll talk a little bit more about how things are transported across the plasma membrane. In terms of the functional analogy, cell is like a car acting. The PM is like walls, doors, windows, and alarm system, determining who comes in and who stays out. And that is the key components of a composite eukaryotic cell. Remember, this is a composite cell. There's no such thing as a typical eukaryotic cell, but a composite cell which helps illustrate key features of eukaryotic cells. And it's understood that it's going to take us some practice to get really good at understanding what all these components do. So we've really just given you a little flavor of the complexity of the cell. And it takes practice, but hopefully the most important thing is you can appreciate how amazing it is that you have 100 trillion of these working in your body, and each one of them has this level of complexity. If you have any questions, please come see me. I'll see you in class otherwise. Have a great day.